Okay, good afternoon. Thank you for joining us this afternoon for the Dana-Farber seminars in oncology. And today's is a special lecture. It's the 2021 Harriet Tischler Memorial Lecture. Harriet Tischler was a former Dana-Farber patient who died of metastatic breast cancer in 1990. And her family established this lecture in her memory to invite leading cancer researchers to share their latest findings with peers and to educate future generations of oncologists. And I could not have more fitting speaker today than my friend and well-known cancer researcher, Ashani Veeraratne from Johns Hopkins. She is a professor and co-program leader there of the Immunology, Microenvironment, and Metastasis program. And she is also a Bloomberg Distinguished Professor at Johns Hopkins University. And many of you know Ashi has been very active in many societies, and particularly the Melanoma Research Society, and has been very supportive of young investigators and women in particular in science. And Ashi is also an expert of the role of aging in tumor progression. Particularly, she's been focusing on melanoma and the role of the microenvironment, aging microenvironment in promoting tumor growth. And today, Ashi will talk about it, her work. The title is Age Against the Machine, How Age Impacts Tumor Progression. Thank you, Ashi, for joining us today.
Thank you so much, Nelly, for the invitation. And I'm also really honored to be giving this named lecture. So I'm very grateful to all of you for choosing me as a speaker. And I guess without any further ado, I'll talk to you about our recent work. And again, if there are any members of the Tischler family, I just want to say thank you again for setting up this memorial lecture in memory of Harriet Tischler. So as Nelly mentioned, she's known me for a very long time, and I think you can tell that because she actually says my last name perfectly, and there's not a lot of people who do that. So today I'm going to talk to you about the work that we've been doing in the space of the aging microenvironment. And the reason we're so interested in age and cancer is because for every 100 people who are diagnosed with cancer, at least 10 of them will be under the age of 49. So 90 of them will be over the age of 50. When you look at people who die of cancer, we can see that the vast majority of deaths occur in people over the age of 50. So there is this clear link between increasing age and cancer. If we look on the molecular level, there are also a lot of similarities that occur between aging cells and cancer cells. If we look at the Hanahan-Weinberg hallmarks of cancer, you can find a lot of these same hallmarks in aging cells as well. And the similarities include things like methylation, genomic instability, metabolism, changes in telomeres and telomerases, as well as senescence. We've been super interested in how the microenvironment itself plays a role in tumor progression. Because I work on melanoma, we've spent a lot of time looking at the microenvironment in the skin, but I'll talk to you about microenvironments in other parts of the body as well. And the reason we're so interested in this is because obviously no tumor is an island. It sits here. Melanomas sit at this interface of the epidermis and dermis. And they're exposed to all kinds of different cell types, from fibroblasts to immune cells to endothelial cells. And our goal is to try to understand the conversations that all of these cells are having with each other and with the tumor cells. We've been very interested in the fibroblasts specifically because the melanoma cell spends a lot of its life right here, becoming invasive and at the epidermis to dermal junction. And fibroblasts create a lot of the structure and the scaffolds that we see in the dermis and a lot of the structure of your skin. And we'll talk a little bit about that later on. And so we wanted to know, since fibroblasts in the skin also have this very unique property of not really turning over, if they're damaged or they die due to age or injury, the existing fibroblasts will kind of stretch out and fill in to fill that space rather than increased growth and proliferation of new fibroblasts. And that's beautiful work performed recently by Valentina Greco in her lab at Yale. So we wanted to know, since this was the case, as fibroblasts age, do they have different properties, and does that differentially affect the way melanoma cells in the skin behave? So what we did was to make artificial skin, and we did that by taking healthy skin from normal donors who did not have melanoma from the upper inner part of their arm to control for the for sun exposure. And we took fibroblasts from young donors in their 20s to mid 30s and older donors in their mid 50s to mid 60s. And we kind of put it all in a dish with keratinocytes and collagen and asked these fibroblasts and skin cells to make an artificial skin. What is fascinating is that when we take melanoma cells and we place them into this artificial skin and we ask what changes, the melanoma cells in skin reconstructs made with young fibroblasts remain tightly nested at the surface of these skin reconstructs, whereas those with aged fibroblasts tend to invade into the reconstructs much more efficiently. And I'll show you some close-ups of that in just a minute. If we also take these experiments and extrapolate them to mouse models using the BRAF P10 mouse model from Martin McMahon and Marcus Bosenberg, that Marcus has backcrossed to the C57 Black 6 mouse model. Marcus has taken these tumors and created a series of cell lines we can now inject syngenetically back into mice. And literally we can take a dish of these cells, split it in half, put half in a young mouse, half in an aged mouse, and then ask what happens. So initially, much to our surprise, what we found happens is that tumors that are implanted into aged mice actually grow far slower than those implanted into young mice. However, when we look at things like metastasis or angiogenesis, we see that tumors planted in aged mice metastasize much more efficiently than those planted in young mice. So recently, Mitch Fain, who is a very talented postdoctoral fellow in my lab, asked the question, is this because the tumors are leaving the primary site at different rates, or are they getting to the metastatic site and not and growing out at different rates? And so what Mitch did was to tag his melanoma cells with M-cherry. He then planted them in the skin of mice and then allowed them to metastasize and took sections serially from the lung. So if he does that at 3 weeks, what he sees is that in the lung, you have these little single cell colonies in both the young and the aged mice. But if he waits just a couple more weeks and he looks at the lungs, what he sees is that those single cell colonies remain in the young mice, but now in the aged mice, they've grown out rapidly, and he can quantitate this over a series of mice. He's done this in these immunocompetent mouse models. He's also done them in nude mouse models, and he sees the same thing quite consistently. So he wanted to know what it was that might be driving this outgrowth in the lung. And so he took fibroblasts from young and aged lungs. He made what we call sandwich reconstructs. So we put in our GFP tagged melanoma cells into collagen and then embedded them with either young fibroblasts or aged fibroblasts and asked how rapidly they grew out. And what he sees is that the GFP tagged melanoma cells proliferate quite rapidly when they're incubated with these aged lung fibroblasts. And this is quite the opposite of what we see in the skin, because if you remember the mouse models I showed you showed that in the aged mice, the tumors did not grow out that rapidly in the skin. And so all of these data combined together make us think that what's happening in the skin is that the cells grow more slowly, but invade better. But in the aged lung, they seem to be growing out far more rapidly. So Mitch wanted to know what it was that these fibroblasts might be secreting that was driving this change in proliferation. And so he analyzed the secretomes of young and aged fibroblasts from the skin and from the lung. And then he compared them. And what he found was that there were distinct changes in the Wnt signaling pathways in young and aged skin fibroblasts, as well as young and aged lung fibroblasts, and that these had almost opposite signatures in the aged skin as compared to the aged lung. So for example, when we look at the aged skin secretomes, we see that there's an increase in non-canonical Wnt signaling, but in the aged lung fibroblast secretome, we see an increase in canonical Wnt signaling, or at least factors that promote canonical Wnt signaling. So I'll just give you a quick snapshot of a couple of these factors. This is a secreted frizzle-related protein. And in the aged skin, we see this this driver of non-canonical Wnt signaling SFRP2. And in the aged lung, we see a driver of canonical Wnt signaling SFRP1. What we have shown before is that H fibroblasts secrete SFRP2, and this inhibits beta-catenin signaling in melanoma cells. It also increases Wnt5a and non-canonical Wnt signaling in the same cells. SFRP1, on the other hand, inhibits the expression of Wnt5a shown by this upper band. And so Mitch asked what would happen if he looked at SFRP1 in aged mice. So if he inhibited this protein in aged mice. So what he did was to take his melanoma cells, he seeded them in the skin of aged mice. He gave them 3 weeks so that they could go to the lung and disseminate. And normally after 5 weeks, when he sections a lung, he would see outgrowth of melanoma cells. But he does this in the presence now of IgG or an anti-SFRP1. And now what he sees is that if he treats the mice with anti-SFRP1 at 3 weeks, he can disrupt this outgrowth of these melanoma cells within the lung. So this was really interesting to us because it fit with something we've seen before, which is what we call the grow or go phenomenon in melanoma, more classically called phenotype switching. And what we see is that melanoma cells grow very rapidly and depend on the Wnt pathway after they've been transformed. And they are guided by canonical Wnt signaling, including beta-catenin, known drivers of melanoma proliferation like MART1 and MITF. However, when they become more metastatic, they switch to a non-canonical Wnt signaling phenotype. And this promotes an invasive and slow cycling phenotype that's also very therapy resistant. And the findings I'm going to tell you about now really sort of turned a lot of what we thought we knew about this process on its head, if you will. So what Mitch did was to talk to our friend and collaborator, Julio Guiso, who is at the Mount Sinai School of Medicine, but moving very shortly to Einstein. And he is a world expert in tumor dormancy, and he has come up with these signatures of genes and proteins that are critical in governing tumor dormancy, and some of them are listed here. What Mitch did was to go to the TCGA database and look at melanoma cells that were either high for Wnt5a or low for Wnt5a to see whether they had these markers of dormancy, because as I said, Wnt5a drives a very slow cycling phenotype. And what Mitch saw is that wherever he had high expression of Wnt5a, he had high expression of genes that Julio has defined as dormancy markers. And the opposite was also true. Where he had low Wnt5a, he saw high expression of genes that we know drive proliferation like MART1, MITF, beta-catenin, and others. So he wanted to know how these different signatures were impacted by the aging microenvironment, and specifically the lung aging microenvironment. So what he did was to take melanoma cells and expose them to conditioned media from either young human fibroblasts or aged human fibroblasts and look at these different markers. And what he saw was that all of the dormancy markers in the same melanoma cells were dramatically reduced by exposure to an aged conditioned media, aged lung fibroblast conditioned media, and markers of proliferation and growth were dramatically increased. So he then wanted to know, since my lab has worked on Wnt5a for quite some time, which is a non-canonical Wnt signaling pathway marker, what the role of Wnt5a might be in all of this. And so Wnt5a is a driver of invasion from the primary metastatic site. So Mitch did this experiment a little bit differently from the experiment he had done with the anti-SFRP1. In this experiment, what he did was to take mice, implant them with the tumors, the YUM 1.7 melanoma tumors that were tagged with M-cherry. And he started docs at 2 different time points. And one set of mice, he started it at day 3 with the idea that he would disrupt the initial seeding of the cells. And in one set of mice, he started it at week 3 to see if it would affect the outgrowth of these melanoma cells, and this was done in young mice. And what he sees is that if he depletes Wnt5a in the young mice, now just like the aged mice, the tumor cells start to grow out much more rapidly, and we can quantitate this over a series of mice. So the reason I say this turned what we thought we knew about phenotype switching on its head is because, for us, we had always just associated Wnt5a with metastasis. And I don't know if there are any trainees in the audience, but I think this is a really important point. I spent the first decade of my career characterizing Wnt5a as this molecule that drives invasion. And I always used to just link it, invasion and metastasis. But the truth is, it's not as simple as that. In fact, Wnt5a overexpression is required for that initial step of metastasis. We think it's required for survival at the distal site. In fact, we're 100% sure of that. But what we do know is that in order for it to outgrow, we need to deplete it. And that's because the expression of Wnt5a is so suppressive to the growth of tumors. So this really, these experiments that Mitch did were really eye-opening for us because they taught us about the complexity of how a gene can drive invasion, seeding at a distant site, and then ultimately outgrowth of metastasis. So this is what we think is happening right now. First, we think there are more Wnt5a positive cells in the aged skin. We've shown that, but regardless, whether young or aged, it's the Wnt5a positive cells that get to the lungs. And these cells can switch back and forth in phenotype from a canonical to a non-canonical Wnt signaling phenotype. However, what we think is happening is that, they're less likely to do so in the young lung because there are factors that keep them in a Wnt5a positive state. In the aged lung, SFRP1 is secreted. It shuts off Wnt5a signaling, promotes a switch of these cells to a canonical Wnt signaling phenotype. Now, this is further exacerbated by the immune microenvironment because in the young lung, the tumor cells are kept in check by having a nice, healthy immune microenvironment. In the aged lung, however, we see an increase in myeloid-derived suppressor cells, an increase in T regulatory cells, and overall a generally more immunosuppressive microenvironment that we think allows for the outgrowth of these cells that are no longer slow cycling, but are now Wnt5a low and highly proliferative. So this is an example of how aging can impact metastasis, for example, and we found that it can have impacts on multiple other factors as well, including therapy resistance, angiogenesis, metabolism, and there's also a lot of impact of aging on the immune microenvironment as well. So I'm going to tell you about a couple of these stories, angiogenesis and metabolism, and then I'm going to give you a quick snapshot of some of the newer directions that the lab is taking. So one thing that we had noticed is that when we implanted these tumors in mice and we stained for all of the different parameters, we noticed that angiogenesis was dramatically increased in mice. And remember that these are the same cells. They're just being injected into either a young mouse or an aged mouse. So, and a lot of this angiogenesis looked like it might be neoangiogenesis. And so we were very interested in how the age microenvironment was affecting angiogenesis in melanoma. We took dermal microvascular endothelial cells, which are the type of endothelial cells we'd expect to find in the skin. We treated them with conditioned media from young and aged fibroblasts. And you can see that when we treat them with conditioned media from aged fibroblasts, we're able to make these nice vascular networks, or these networks of endothelial cells. This was a little bit confusing to us, though, because one of the things that we had seen was that VEGF, which is obviously one of the key drivers of angiogenesis, especially in tumor formation, its isoforms and its receptors were all decreased in melanoma patients as they got older. So this didn't really make a whole lot of sense to us because we were seeing this dramatic increase in angiogenesis, but then a decrease in the factors that should be driving it. However, we weren't puzzled for too long because if you remember SFRP2 that I mentioned, which helps to drive invasion, It's also been shown to stimulate angiogenesis, and it does this via calcineurin and FAT signaling pathway. And this is work out of Nancy Klobodimor's lab that was performed in breast cancer. We had identified SFRP2 in the secretome of our aged skin fibroblasts. And we also knew that if we treat animals with recombinant SFRP2, they will have an increased metastasis to the liver. So we knew SFRP2 could drive invasion in our models. And now Mitch, Aman, and Brett, who were in the lab at the time, decided to explore whether it could also affect angiogenesis. So they took the dermal microvascular endothelial cells, they treated them with the age conditioned media, saw that it increases vascular networks, just as I showed you before. And the same is true that if you just give those endothelial cells recombinant SFRP2 in the presence of young conditioned media, or if you treat aged fibroblasts with an antibody against SFRP2 and then put that on the endothelial cells, now they no longer are able to form those vascular networks. We can do the same experiment in mice. And so what we see there is that, again, the exact same thing. If we give young mice recombinant SFRP2, the tumors have more angiogenesis. And if we give aged mice an antibody against SFRP2, we can disrupt angiogenesis in those mice. So that was all. And I should also mention that in young mice, the tumors expressed a ton of VEGF, but not in aged mice. And the opposite was true for SFRP2. So that was all super interesting, but we couldn't help but think, what did that mean for patients who are treated with Avastin, which is an antibody, I'm sure, as you all know, directed against the VEGF receptor and intended to target angiogenesis in cancer. It's in clinical trials for multiple cancers. It's actually approved for the use of some cancers. And it turns out that a group in the UK did this huge trial called Avastin, where they looked at their over 1,300 patients with metastatic melanoma, and they gave them Avastin they gave them Avastin as an adjuvant therapy and looked at their risk of recurrence and overall survival. So there was no change in either of these. And so what we did was to reach out to Pippa Corey and Mark Middleton, who were the lead investigators on the study, and asked them if they wouldn't mind going back and reanalyzing their data, and this time looking at age as a parameter. When we do that, we see that there actually is a difference. If patients are under the age of 45, they will actually respond to Avastin, whereas patients over the age of 65 do not respond to Avastin. So this was in keeping with our data that we're targeting VEGF in these older patients, but there's not much VEGF to target. So we wanted to show also that in the presence of SFRP2, targeting this VEGF would be useless. And that's exactly what we see here. If we look at tumors in young mice, if we treat those with an antibody against VEGF, we can decrease angiogenesis. However, if we try to do that in the presence of recombinant SFRP2, which dramatically increases angiogenesis, treating with anti-VEGF does nothing to the level of angiogenesis within those tumors. So what we've learned from these data is that during aging, while angiogenesis increases, VEGF and its isoforms and receptors decrease, SFRP2 increases, and this drives angiogenesis. It's actually a very potent driver of angiogenesis. It's able to inhibit the efficacy of anti-VEGF in vivo, or at least, it overcomes whatever effects anti-VEGF might have. And our data from patients show us that younger patients fare better on Avastin than older patients. We have also recently become interested not only in the number of the blood vessels being produced, but the quality of the blood vessels. So the matrix changes that occur during aging might make blood vessels more permeable. And this was based on some work that we had done looking at the matrix during aging. So this is another one of those skin reconstructs, again, with this melanoma cells in the young remain at the surface of the skin reconstruct, but in the age they start to invade quite dramatically. The only thing that is different between these two skin reconstructs is the age of the fibroblasts that we use to make them. But nonetheless, you can see if you do 2-photon microscopy on these young and age skin reconstructs, the density of the collagen and the arrangement of the collagen is quite different. If we look at a normal mouse skin that does not have tumor in it, we see the same thing. We see that the collagen is very dense in the young mice. In the aged mice, there's less collagen and it's more organized into these fibrils. So Aman in the lab was curious as to why this was. And so she looked at the secretome again, and the protein that was most increased in young fibroblasts was this protein called haplin-1, which stabilizes aggregates of proteoglycan monomers. It basically takes hyaluronic acid, it binds it up with collagen and elastin and acts as a knitting needles that maintain this dense structure of collagen in the skin. So if she takes aged mice, which remember have all of this sort of more organized collagen, she treats with haplin-1, she can start to knot that collagen back up. And so what Aman did was to ask what this meant. Actually, I can show in the summary diagram. What she asked was what this meant for the invasion and metastasis of melanoma cells. And she finds that if she looks within the primary tumors in young and aged mice, the loss of haplin-1 results in the degradation of the ECM, this alignment of collagen, less cross-linking, and this has impacts not only on the escape of tumor cells from the primary site, but also the infiltration of immune cells. And so we're still trying to dissect some of that out a little bit. At the same time, in a parallel study also published in the same journal, Brett Ecker in my lab was very, very interested in looking at lymph nodes and metastasis to the lymph nodes. And the reason he was so interested in that is that in melanoma, lymph node metastases are actually far more common in young patients than in aged patients. But aged patients die of visceral metastases far more frequently than young patients do. And so he wondered if the ECM around the lymphatic vasculature, as well as the lymph node, might be impacted by aging. And part of why he wondered this was because he had noticed that when he tried to do sentinel lymph node imaging in patients, he would see that the dye would not get to the lymph node, even in the absence of cancer, very efficiently. So when Brett looks at the matrix around the lymphatic vasculature, he sees that in the young, it's very nicely organized and quite dense, but in the aged, it starts to break down, and a lot of this is due to haplin-1. So basically, haplin-1 is playing a role, not only in maintaining the integrity of the dermal ECM, but also the ECM that provides some structure to the vasculature within these tumors and the vasculature that acts as conduits that leave the tumor. So the loss of haplin-1 and ECM integrity may not just affect the invasiveness of the cells themselves, but also direct the route of metastatic dissemination during aging. So Gloria Marino in the lab was very interested in this, and she wanted to ask what it meant for actual blood vessels and angiogenesis. And so what she did was to image blood vessels in young and aged skin, and just again, a reminder what that looks like, what the skin looks like in the absence of anything, of any tumor. And what she sees is that blood vessels in the aged tumor microenvironment sit in this very low collagen environment, whereas those in the young microenvironment have a lot more collagen around them. She wanted to know what this meant for the integrity of those blood vessels. So she took endothelial cells and she took young fibroblasts, and she asked those young fibroblasts to lay down a matrix. And on top of that matrix, she seeded these endothelial cells. And her hypothesis was that the endothelial cells are connected to each other by V cadherin, but these connections are stabilized by their connections through integrins on the matrix. That being the case, she wanted to know if when we then seed the same cells on each matrices, would these interactions between the cells and the matrix be disrupted, and would that in turn disrupt the V cadherin junctions? And so that's exactly what she saw when she seeds endothelial cells on young matrices. They have nice V-coherent zippers. When she seeds them on age matrices, the intercell staining disappears. And then she asked, what does haplin-1 have to do with it? Can it tighten up all and cross-link all the matrices? So the first thing she did was to deplete haplin-1 from the young fibroblasts, and let them make their matrix, and then seeded cells on top. And now she sees that that looks just like the aged fibroblasts, and they've lost this ability for the V coherent to make these connections. The opposite is true if she adds in recombinant haplin-1 to her aged ECM. Now she can knot these matrices back up, and the V coherent staining appears beautifully between these cells. What does that mean? Well, that means that that's probably affecting barrier integrity. That means flow in and out of the vasculature. And so what Gloria did next was to collaborate with Karen Eisinger Matheson at Penn. She seeded endothelial cells on her ECMs and put those on electrode arrays. And then she ran a current through the electrode and asked, do the cells resist? And so the idea is, as cell density increases, resistance through these cell-cell junctions will increase. Because there is barrier integrity. So if she seeds cells on gelatin as a control, they have plenty of barrier integrity. It increases over time. If she seeds them on a young matrix, a young fibroblast-derived matrix, it does the same thing. It increases over time. However, if she seeds them on an aged fibroblast matrix, there's very little increase over time, and there seems to be very little resistance whatsoever. But now she treats those aged fibroblasts with haplin-1, has them make a matrix, seeds the endothelial cells on top of that, she can reconstitute that barrier integrity. So that tells us then that endothelial cells seeded on an aged, that endothelial cells in an aged microenvironment probably have a lot less barrier integrity and weaker cell-cell connections. So she wanted to see that in vivo. So she took tumors from mice, either young or aged mice, that have been treated with recombinant haplin-1, and she looked at the V-coherent staining of the endothelial cells and vessels within these mice. What she saw is that the vessels, which you can barely see here in the aged mouse, were much less positive for V coherent than the vessels that are in the young mice. And if she treats the aged mice with haplin-1, she can again increase that V coherent staining. So what she saw in vitro pans out in vivo. However, I should point out the fact that there are actually far fewer blood vessels in the young mice and in the young plus haplin-1. And that's what this slide will show you. So if she first does this in vitro using endothelial cell spheroid assays, she sees that aged mice have more protrusions of these endothelial cells, telling us that they are likely to make tubules. The same endothelial cells seeded with young fibroblasts are less able to do that. And if she adds haplin-1 to the aged, they decrease their ability to make these protrusions. And if she treats the young fibroblasts with a haplin-1 knockdown first, they now increase their ability to make these nice protrusions. If she takes this experiment in vivo, she sees the same thing. So in vivo, the tumors in aged mice, as I've shown you a few times now, have far more blood vessels than those in young mice. And if she treats them with haplin-1, she can actually decrease the amount of blood vessels. So that was a little surprising to us. And that's a finding we're still working on to try to figure out, because we didn't expect haplin-1 to have an impact on the number of blood vessels, just on the integrity of them. So now Gloria is trying to figure out how haplin-1 might be affecting angiogenesis, even, as a process. All right, so I will switch gears one more time and tell you about our findings of how these cells change metabolism as they age. So Gretchen Alicea, who's a postdoc in the lab, she also did her PhD with me. She decided to look at metabolism that was changing with age and how that impacted tumor cells. And the reason she was so interested in this was that she saw that young fibroblasts in whether they were, I'm sorry, that age fibroblasts, whether they were in a low glucose environment or a high glucose environment, had a lot more lipids that they expressed than young fibroblasts. And that's because, to take you back to your Krebs cycle and all of that, the cells take up glucose. Glucose gets processed through a couple of different pathways, but ultimately ends up making fatty acids. And so what she found was that in a higher sugar environment, older fibroblasts would take up a lot more sugar than the younger fibroblasts and use that to make the lipids. And if she blocks this transporter of glucose, she finds that she can dramatically decrease the production of lipids in the H fibroblasts, even in very high sugar conditions. She wanted to know what that meant for melanoma cells. Were they secreting these fibroblasts? Because if you look at these images, it looks like the lipids are just everywhere. And so she took her melanoma cells, she treated them with conditioned media from either young fibroblasts or age fibroblasts, and found that when she does that, the melanoma cells have far more lipids when they're treated with conditioned media from age fibroblasts. She did a bunch of lipidomics. She found that the same lipids that were increased in the age fibroblasts were the same ones being taken up by the melanoma cells when they were exposed to that conditioned media. And she also showed that if we take fibroblasts and we stably label them with Bodipi, wash that Bodipi stain off, and then co-incubate those cells with melanomas that are GFP tagged, what she found was that, again, in the presence of the H fibroblasts, the melanoma cells would take up the lipids. And so what you're about to see in the images I'm going to show you, wherever you see a green cell, that's a melanoma cell. Wherever you see a red cell, that's a cell that has lipids in it that could only have come from the fibroblasts. So with her green melanoma cells co-incubated with young fibroblasts, she sees that there's a low level of uptake of lipids from these fibroblasts. However, those same cells incubated with H fibroblasts take up a ton of lipids. And you can see that there are even cells that have these green nuclei and completely red cytoplasm. And almost every single cell is taking up lipids from these H fibroblasts. So she wanted to know how that was happening. And so she looked at a series of fatty acid transporters in her melanoma cells. None of them really changed with age, except for this fatty acid transporter, FATP2. And FATP2 expression is increased in melanoma cells exposed to age-conditioned media. It's increased in skin reconstructs made with age fibroblasts, and it's increased in our YUM 1.7 tumor model, in the tumors implanted in age mice. And of course, it's increased in patients and melanoma patients as well. So Gretchen then inhibited FATP2 in her melanoma cells, exposed them to conditioned media, and showed that they're no longer able to take up lipids. She also treated with a drug called lipofumata, which targets both FATP2 and FATP1. And she looked at the lipid accumulation in the presence of lipofumata. And again, by inhibiting FATP2, she was able to inhibit the uptake of lipids. So the next experiment she did then was to see how this could impact targeted therapy. And the reason is that because we know that altering metabolism in melanoma cells can dramatically impact their response to therapy. The therapies that we use for melanoma, in addition to immunotherapy, are therapies that are targeted against the BRAF oncogene and its signaling pathway. And what Gretchen had and what we had shown before was that older patients are less likely to have a good response to targeted therapy than younger patients. And that was really surprising to us because we kind of thought of this as a one gene mutation to which we had a drug. And so it was a one gene, one drug, and nothing else should matter. But even doing that experiment in genetically identical cells in two different microenvironments, there was a very different response. So here's an example of that. If we take our YUM 1.7 model, we implant it in either young mice or age mice. Again, they grow better in the young mice. When we treat with a drug targeted against the BRAF oncogene, those tumors regress in the young, but they don't really respond at all in the aged. And so when we look at patients that respond to the drugs and those that don't, what we find is that patients who do not respond to the BRAF inhibitors, they have far more positive staining for this FAP-P2 marker. So what Gretchen did next was to knock down, to create an inducible knockdown of FAP-P2 in her YUM1.7 cells. And then what we did was to implant these cells on either young mice or aged mice, and then treat with the BRAF MEK inhibitor, either in the presence or absence of FAP-P2. And this is really one, I think, of our favorite experiments that we've ever done, because we saw some things very dramatic. First of all, the green line here is just the empty vector control. But if you look at the blue line, this is just the BRAF MEK inhibitor. And in the young, the tumors respond to the BRAF MEK inhibitor, they go away for a while, and then they come back. And this is completely standard and expected. If we deplete FAP-P2 in these young mice, nothing happens. It stays exactly the same. Now, if we do that exact experiment in aged mice, it's a completely different story. So first, treating the aged mice with a BRAF MEK inhibitor only slows down the tumor for a little bit, and then it continues to grow. It never really regresses. However, if you now decrease FAP-P2 in these mice, in these tumors, in combination with the BRAF MEK inhibitor, not only do those tumors regress, they stay gone for a really long period of time. So that was a very exciting experiment, because it told us that you could really target age-related changes and have an impact on therapy. So I'll end this talk with some of our newer data. So in addition to age, we're now looking at sex. And that's because melanoma, like many other cancers, in fact, has a difference in the incidence and the rates of cancer as we age in men versus women. So Yash Chhabra in my lab is studying what we call our Venus-Mars project, based on this book from the 80s. If you guys remember, Men Are From Mars, Women Are From Venus. And basically in melanoma, we know that survival is different in women versus men. Things like TIL infiltration is different in women versus men. But what Yash has been finding is that what one of the critics of this book said back in the 80s is that the view that men and women are so different that they can't understand each other is a bit extreme. And I think Yash is finding exactly the same thing, where he sees that while the magnitudes of difference between male and female fibroblasts and their impacts on tumors differ, the trends do not. So if you look at stuff like senescence, for example, you see that female fibroblasts, both male and female fibroblasts increase senescence as they age. Female fibroblasts start out already a little more senescence, and then they increase their senescence. If we look at things like exosomes, for example, so the secretome of fibroblasts, again, young and aged, male versus female, Laura Hughes in the lab has shown that in aged male fibroblasts, there's a lot less CD9 secreted in the exosome. There's a decrease in aged female as well, but obviously it's nowhere near as dramatic as what we're seeing in the CD9 expression in exosomes in aged males. There's metabolism differences between males and females in things like the secretion of extracellular lactate, in things like ROS, in things like mitochondrial ROS. And so Yash has shown that, again, there's a more dramatic impacts between male young and aged versus female young and aged. If we look at the impact of those fibroblast changes in male and female on melanoma cells, again, we're seeing the same thing. So this assay is a spheroid assay. And what we're looking at is are these spheroids of melanoma cells that have been treated with conditioned media from either young males or aged males, young females or aged females, and then in this row treated with the BRAF MEK inhibitor. So normally in the presence of the young conditioned media, or no conditioned media, melanoma cells will die when treated with the BRAF MEK inhibitor combination. However, in the presence of age conditioned media, they die less. And in the presence of age female conditioned media, they die even less. So again, although female conditioned media is protective, it's more protective than male conditioned media. I hope I'm making sense. And finally, we're moving beyond melanoma. I've been collaborating with my good friend Liz Jaffe and Dan Zabransky, who's a very talented oncology fellow. And what Dan has done is he's obtaining young and age pancreatic fibroblasts from healthy patients, again, doing a lot of the similar assays that we've done in melanoma, proteomics, analysis of previously established targets, and understanding their response to therapy. He's also looking at this in mice, and I'm going to show you that data in just a second. And we're trying to characterize the pancreatic microenvironment in young versus age mice and the impacts of that on biophysical changes, metastasis, etc. So using the KPC model, what Dan has done is to take 64-week-old mice compared to 6-week-old mice, orthotopically inject KPC cells, and then image these mice over time. And I think that what you can see quite clearly is that the age mice develop tumors far more quickly than the young mice. This is a tumor weight, and I will tell you that we have now examined metastasis, and the age mice have far more metastasis in multiple different metastatic sites than the young mice at the same time point. So we're very excited by these data, and Dan is preparing to do all of the molecular analysis of these. So I hope that I've managed to convince you that the aging microenvironment is absolutely a critical driver of metastasis, whether it comes to biophysical changes in the matrix, angiogenesis, metabolism, or metastasis. And we kind of need to really think about how we're designing both preclinical and clinical studies. These are all of the amazing people who do the work in the lab. Our funders, I've tried to call each of them out as I mentioned their work, but they really are a team and we work beautifully together. Collaboration is a global effort. Many of my collaborators are right there at Harvard, they're across the street and across the world. I love to usually end my talks with a picture of my favorite aging study, who Nellie has known since, well, before she was born even. And when I was telling her today that she was driving, has a job and a boyfriend, she couldn't believe any of that because I think she remembers her best from this age. And finally, I will give, as I always do, a shout out to the fact that we need to consider diversity in academia of the people who get STEM degrees. Women of color make up a very small percentage. And if you are really interested in increasing racial diversity in the spaces in which you exist, I encourage you to use the resources we put together in this article with my friend Anita Brady in Cancer Discovery. There's a lot of good websites and links in there to help. And so I will stop there and take any questions.
Thank you, Ashley. That was amazing, really impressive and really nice talk. So while we wait for some questions from the audience, I do have some question, and related to the last issue that you discussed, are there any ethnic differences? Because as you know, there are a lot of skin disorders, and wound healing is very different in the different ethnic groups. So that would, I assume, influences matrix and all that. So have you looked at it?
So we are just beginning to look at it. What I will tell you is melanin is obviously a very important factor in melanoma. But what's interesting is that people who get melanoma, people of color who get melanoma tend to get largely acral melanomas rather than cutaneous melanomas. So Vito Rebecca, who's a new faculty that I recruited to Hopkins, he is doing some beautiful work on acral melanoma. We are very interested in the mechanical changes that happen with age in the skin, as I mentioned. And we think that those might be driving some of the changes in acral melanomas. So does race impact the actual onset and progression of cutaneous melanoma? That is more unclear to me. I almost think it would be more protective, because if we think about things like UV The damage that UV is causing, it looks very similar to, I didn't present that data, but we've looked at the secretome of UV-induced fibroblasts compared to aging fibroblasts, and there's a lot of similarities there. So I would imagine melanin would be protective of that. But it does impact what types of melanomas patients get. And we think that those can be impacted differentially then by aging.
Okay, so one question from Ed Frisch here. Any relationship of aged and young fibroblasts to other CAF phenotypes defined in the TMA? As you know, like there were these single cell studies now, and there are many different fibroblasts. So have you looked at that? I mean, any particular one more or less affected by age?
So, you know, we have not looked at it in depth. There was a great review on fibroblasts that we put together from a Cold Spring Harbor meeting. One of the amazing, great late Zena Werb was an author on that. And we talk about all of these different populations, and then we talk about aging. So what I will tell you is that aged fibroblasts, a lot of people ask me about senescence and SASP. While there are similarities, they're not identical to senescent fibroblasts, and they look a lot like CAFs, and they look more like MyCAFs. Have we done the exact profiling to say that definitively? No, but I can tell you just from my experience, there's a lot of similarities.
Another question is related to the, the metabolic differences that you showed. So does the dietary restriction remove differences in tumor growth between young and old mice? Because, like there's this anti-aging diet that if you don't eat, then you don't age. So would that alter the tumor growth? Or I mean, diet in general, like, ketogenic diet, so to influence response to even chemotherapy in many cancer types?
Absolutely. And so great question. There is a lot of data out there on calorie restriction and tumor growth. And so, one of the experiments we tried to do unsuccessfully, not because, it was sort of at the time where I was leaving Wistar, coming to Hopkins, and then the pandemic hit. So we've not repeated it again, but it was more technical difficulties, was to try to do the same experiment that we did for the FAP-P2 inhibition. So young mice, age mice, BRAF MEK inhibitors, plus minus calorie restriction. So we think that that's going to impact it. But I don't know for a fact that it will. We had some changes. I'm sorry, my dog always runs at this time. She likes to tell me when she's had enough.
Okay. So there are two more questions here we can ask. Is like, oops, popping up. Did you see changes in EMT and MET programs in melanoma cells grown in young versus old? I think so actually that's a lot of the dormancy stuff I was talking about. Those programs, AXL, MITF, all of those in melanoma are involved with, melanoma doesn't do it classical EMT, but they do become more mesenchymal. And so what we see is that in a situation where you have aged skin fibroblasts, you see a more mesenchymal phenotype, but where you have aged lung fibroblasts, you see a more epithelial. And so that really defines that phenotype switch from the leaving that primary site, going to the metastatic site. You have a EMT and then an MET in the distal site. Okay, and the other question is, how do you view the complex relationship of aging and cancer stem cells and the aging and the microenvironment?
I view it as complex. So it is interesting. So the notion of the cancer stem cell in melanoma has been under debate for quite some time. There are definitely stem cells that reside in the hair follicles. So that is going to be super interesting. We have not explored it, and I'm not sure anyone has explored it really in melanoma. So give me a couple of years and maybe I'll actually have an answer to that question.
Okay. And another question is like TGF-beta, as you know, I mean, the angiogenic phenotype that you saw and those differences in aging is also very interesting. And, like TGF-beta polymorphism I know influences breast cancer risk. Yeah. And that could be because through the, many things, but the microenvironment. So have you looked at are there any kind of polymorphisms in genes that would influence any of this age effect and then melanoma? Like
Yeah, I mean,
TGF-beta, for example, TGF-beta pathway.
yeah, so taking TGF-beta specifically, which we have looked,
Yeah.
we do see a switch from beta-1 to beta-2 young to age in the lung microenvironment. And we think that's part of what's driving these cells as coming out of dormancy.
Okay.
So there is a very definitive switch between one isoform versus the other, which is also what we're seeing in the Wnt signaling pathway.
Mm-hmm. But they're supposed to bind the same receptor. So how do you get the different?
Yeah, so that's actually a really interesting question. So looking at, for example, AXL and the MER tyrosine kinase, I didn't present that data either, but we have a paper under review on that all of that. It's very interesting. So, AXL and MER-TK both bind Gas ligands and pro-S1, but they do very different things once that happens. So I wonder whether the reverse is happening with TGF-beta 1 and 2, where if it's TGF-beta 1 binding the receptor, there's a different signaling pathway influenced by the microenvironment than TGF-beta 2. And in melanoma.
I actually just have an idea. It's maybe crazy, but TGF-beta 2 is more requires the beta glucan TGF-beta receptor 3 for binding the receptor.
Okay.
So I mean, despite the fact that the signaling receptor is the same, the way they present the ligand is different. And IR3 is a proteoglycan, so it may depend more on the matrix and some of those hapline and whatever you were showing about the aging, because that could influence the distribution. So that could be something interesting to look at, because
that's a great idea.
In the breast, we also see that the 1 and 2 very differently expressed in different cells. So I mean, I don't know. And then the receptor 3 tends to be differentially expressed. So that could be interesting to look at. But
super
anyway,
interesting.
I think if there are no other questions. There's one more question. I'm sorry. We have seen in aged breast that endothelial cells show dramatically decreased expression of genes associated with tight junction and adherence junction, as well as lower NCM expression in the fibroblast. Based on your study in the skin, do you think the later is the cause of the former?
I think it would definitely have an impact.
Yeah,
I mean, I would have to say definitely, right? If the ECM around them is changing, clearly there's going to be, as I showed you, a disruption of those cell-cell interactions. So yeah, absolutely.
Okay. All right. Thank you so much, Ashi. This was a really nice talk, and lots of other questions we could ask and look at aging, and looking forward to the future studies.
Okay. Thank you so much. Take care, everyone.
Bye. Bye-bye.
Bye.